Hello. Welcome to the session on microbes, diet, and immunity. My name is John Hamilton. I am a science correspondent at NPR, formerly National Public Radio, now just NPR. I'll be your moderator for the next 50 minutes. So my own introduction to the microbiome happened about 12 years ago. I was doing a radio story about sushi, specifically maki sushi, which has seaweed in it. And one of the things I learned was that people from Japan, the island of Japan, can digest the seaweed that is in maki sushi. Other people like me from the mainland cannot. And I assumed that the reason for that was because, well, their genes must have changed. They must have adapted somehow to be able to digest this. But the story I was doing was about a study that found, no, that is not the case at all. The reason that people from the island of Japan are able to digest the seaweed in maki sushi is that the microbes, the bacteria in their gut, that's what has evolved. It's that evolution that makes it possible. And so that got me thinking, what else might the microbiome be involved in? And of course, it turns out the answer is just about everything. You know, these trillions of microbes that live in our bodies have a huge impact. It's changing medicine, the way we think about diet, our understanding of the immune system. It may even have implications for diseases like cancer, disorders like Alzheimer's, and autism. So today, we're going to hear from two remarkable scientists who are expert in this field. Val McKenzie is Associate Professor of Ecology and Evolutionary Biology at the University of Colorado Boulder. She knows a lot about the microbiomes of wild animals, including fish, amphibians, bats, and a range of vertebrates. She's also someone who is working to diversify her field. She's launching a program for graduate students from a wide range of countries and backgrounds. And the idea is that they will spend a month in Boulder doing field work and getting a chance to meet other young scientists. And it will cost them nothing. So kudos for that. Rob Knight is founding director of the Center for Microbiome Innovation and a professor of pediatrics, bioengineering, and computer science. You really do all that. And engineering at UC San Diego. His research has linked microbes to many different health conditions. He also runs a program that detects the virus responsible for COVID-19 in wastewater. And he's written 2 books about the microbiome that are meant for the general public. One is called Dirt is Good. The other is called Follow Your Gut. So each of our scientists is going to give a presentation. Rob, let's start with you.
All right, great. Well, thanks so much, John, for that wonderful introduction. And what we're going to talk about today is the beautiful dance that links microbes, diet, and immunity. 3 topics that you might not think to go well together, but by the end of this talk, I hope you'll be thinking differently. Was that advancing? No, you can't see that on the big screen, can you? And it's still not up there. There we go. Great. So there's a lot of revolutionary discoveries coming through in microbiome science at the moment, as you probably read about in the news, heard on NPR and so forth. And what's fascinating is all this work is starting to change how we see ourselves as human. So I'd like you to start by just taking a moment to reflect, if you will, on what you saw when you looked in the mirror this morning as you were getting up and getting ready to come to this wonderful festival of ideas. For myself, I saw an organism that's just 43% human, and not just because it was early and I hadn't had my coffee yet, but if we consider our bodies at the level of cells, each of us has about 30 trillion human cells, according to the latest estimates. That's a lot. But we have about 39 trillion microbial cells, which is even more. And so that's where that 43% human number comes from. Even at the level of cells, those microbes, mostly bacteria in your gut, but also a whole bunch of other stuff and all over your body, makes up more than half the cells in your body. Now you might think, well, who cares about cells anymore? It's the 21st century. We should be thinking about DNA. And when we think of it that way, it's even more remarkable. So you probably remember from high school biology about your human genome, where half comes from your mom, half comes from your dad. When we think about all of those human genes as mapped by the Human Genome Project, we've got about 20,000 of them. But amazingly, when we look at the microbial gene catalog, we have 2 to 20 million microbial genes in that microbial gene catalog. So by that measure, counting up the unique genes, you can say we're at best 1% human. And what's most remarkable about this is for all the money that's been spent mapping the human genome, you could say that we're ignoring 99% of the system. And what's more, the ones we're ignoring are the ones that we can change. So each of us has changed them profoundly through our lifetimes. We continue to change them. And what's coming is getting a much better sense of how we can change them to maintain and improve our health over our lifetimes. And whether we want to change them or not, as you heard from John, they're changing automatically, picking up genes from our food even, from other bacteria we're exposed to. And that's an ongoing process that happens whether we want it to or not. Now, I said most of these bacteria are in the gut, and in this respect, it's important to remember that the gut is not Vegas, and that what happens in the gut doesn't stay in the gut. And things that go on in the gut affect all of our systems all through our bodies, as we're just learning. And these include positive effects, including educating our immune systems, helping the brain develop normally, even helping in fitness. So elite athletes have different microbiomes from people who are more sedentary. But it also includes a lot of negative effects, including things like gastrointestinal disorders and metabolic disorders, like you'd expect, but also diseases of the liver, of the heart, even the brain. So we have a large project going on Alzheimer's disease at the moment, and looking at how changes in diet can mitigate Alzheimer's disease by changing the microbes and the microbial genes. So, that's the impact of the microbiome on our health. We also have a lot of impacts on our microbiomes. So the microbiome records a lot of data about our lives, starting from how we were born, where how we're delivered has a huge impact on our first microbes, and even into adulthood, where they're linked to autoimmune disease. But also our diet, our exercise, whether we're aging successfully or unsuccessfully, the drugs we're taking, even where we live, has a huge impact on those microbes, where we can read out their state by looking at the DNA from a stool sample, or from a skin swab, or from some other part of the body. Now, it's especially important that we study them now because we're going into a microbial biodiversity crisis. And I'm sure you're all familiar with Rachel Carson's work, where she documented how DDT and other pesticides were wiping out all kinds of things in the ecosystems around us, like the birds, for example, no longer singing in the spring. And my good friend and colleague Marty Blaser at Rutgers has documented this for our inner ecosystems in his wonderful book, Missing Microbes, that he published a few years ago, where he goes into not just how antibiotics, which is what made the cover, but also things like low-fiber diet, lack of exposure to natural microbiomes around us, and even things like caesarean sections are reducing our microbial diversity within us. And what's amazing about this, and this is one of Marty's favorite graphs that he likes to show, is over the 20th century, where one disease after another was brought under control by advances in public health, everything from measles to TB. At the same time, you see this explosion of things like asthma, type 1 diabetes, multiple sclerosis, Crohn's disease. And when this was published 20 years ago in one of the top medical journals, we knew those diseases were linked to the immune system, but we had no idea that they were linked to the microbes. Whereas today, we can cause or cure analogs of these diseases in mice by modifying their microbiomes, and they've all been linked to the microbiome in humans. So, another reason for concern is that when we study people living much more traditional lifestyles, like the Hadza hunter-gatherers, what we see is that they have a lot of kinds of microbes that we've lost, and these are the last hunter-gatherers in the Great Rift Valley in Tanzania, where the birthplace of humanity, still foraging 95% of their calories from the wild. And they have a tremendous amount of seasonal cycling in the microbiome that we see greatly reduced in people living more industrialized lifestyles. And when we look across human populations, they automatically sort out from the least industrialized to the most industrialized by their microbiomes. And this is coupled to going from a much more diverse microbiome state with entire major groups of bacteria like the spirochetes disappearing and being replaced by a much less diverse community in North America, in Europe, in Japan, and in other countries that are more industrialized. So it's almost like we took this amazingly rich inner ecosystem and bulldozed it and replaced it with a cityscape where only the rats and the pigeons survive. Now, one thing that's of tremendous concern with respect to the link between microbes and diet and health is cancer. And this is Greg Poor, who recently graduated from our MD PhD program at my lab, And he was fascinated by the fact that our environmental studies, like in the Earth Microbiome Project, had seen microbes everywhere from the upper atmosphere to the rocks deep in the earth. But people thought of cancer as a completely sterile entity with no microbes. And the question was, how could microbes possibly not live there, given that they live everywhere else? And there's these classic papers called the hallmarks of cancer papers that tens of thousands of studies have been based on, but they don't even mention microbes in cancer. So Greg was able to find, using advanced computational approaches, and this thing called the Cancer Microbiome Atlas, that in fact microbes linked to tumors throughout the body. But he was able to find, even more remarkably, after correcting for technical variation, that you could do it that you could do it not just from the tumor, but from a blood biopsy. And the reason why Greg succeeded where others had failed, this is a little bit technical, but the state of microbiome studies at the moment is that you can't compare between different labs. So if you look at the picture on the left, you can see that all the different samples cluster by what lab did the work. Whereas after Greg applied these machine learning techniques, you can see that all that variation went away, and what was left over was information about the cancer and about the patient. So to cut a long story short, this is just to give you an idea that this is backed by science, not just by rhetoric. But Greg was able to show that not just the microbes out of the cancer, but microbes out of the blood could tell what cancer an individual patient had, and also what stage it was at for many of those cancers, with up to 99% accuracy. And he's currently founded a company to commercialize this technology and get it out of the lab and into the clinic, where it can really help people. Now, Jennifer Wago, who's a professor at the MD Anderson Cancer Center, has taken this a step further, not just for diagnosing cancer, but for curing cancer. And immunotherapy by checkpoint inhibitors, one of the latest cancer treatments, she's found that whether it works for you depends tremendously on what kinds of microbes you have. And most recently, she's shown that through the combination of prebiotic fiber in the diet and probiotics, adding in good microbes, she can actually move people from not responding to that therapy to having it save their lives. And so this is a tremendous demonstration of the impact of the microbiome on cancer to cure it. And one of the world's leading, and so the goal here is for microbes to improve each stage of cancer care, from diagnosing what type of cancer you have, to predicting what's going to happen later on based on the microbes you have, to therapy, modifying what drugs you're going to take so that they'll be safe and effective for you, and then checking that you're still in remission. And one person who's benefited tremendously from this is one of the leaders in the microbiome field, Norm Pace, who's recently retired as a professor from CU Boulder. And in 2015, he was diagnosed with stage 4 melanoma, and essentially everyone had written him off as a goner. But his microbiome turned out to be just right for Keytruda, one of the first immunotherapies to hit the market. And he recovered miraculously from it through this coincidence that his own microbiome, which we'd studied in various projects, turned out to be just right for this therapy. So our vision with this is to take it out of the lab and get it into your homes. And the idea is that as you view yourself in the mirror in the morning, maybe we can modify that so it's a display screen that can tell you about your microbiome as well as showing yourself, where the idea is that we could maybe take a sample of your breath, look at the products of microbial metabolism in your breath. Obviously, we're still working on the user interface for this part, because you probably don't want to see this first thing in the morning unless you really loved organic chemistry. But the idea is to translate it into the kind of microbiome profile that we give people in our citizen science projects, like the American Gut Project, and then ultimately translate it into where you are on a microbiome map of other healthy and unhealthy people. So we can show you if you're at risk for anything, and then either tell you to consult a physician, or alternatively tell you something you can do yourself with consumer products like probiotics. And so while we're dreaming about this, your smart mirror could potentially connect with your smartphone over the Internet of Things, so that when you're faced with 1,000 yogurts in the supermarket, you can zoom in on the one that you actually need that's got the right organism in it, scan its barcode to confirm that you got it. And then our dream is that your smartphone would also communicate back to your mirror and tell you about, so that it could take all the things that affect your microbiome, like what you ate, how much you exercised, how many minutes outdoors, which also has an impact, what sort of space you inhabited, who you were with, and then shows yourself visions of the future by modifying your image in that display screen to show yourself 5, 10, 20 years down the track what you'll look like if you behave the same every day, or what you might look like if you do a little better or a little worse. And many of the components, including things like the AI to modify your image in real time, already exist. It's just a matter of finding the data and training the models so that we can do those predictions accurately. So that's the kind of crazy stuff we're up to at the Center for Microbiome Innovation that I direct at UC San Diego, with a growing list of corporate partners and over 100 faculty members involved, who we're incredibly grateful to, as well as a huge number of collaborators on other projects, the many people in my lab who it's a true pleasure to come in and work with every day, and the tens of thousands of members of the public who've contributed to our citizen science projects. So with that, I'd just like to say thanks. We'll answer your questions together in the discussion at the end of this, and I'll hand things over to Val, who's going to tell you about the other piece of this puzzle.
All right, so Val is going to tell us about the microbiome and the environment and wildlife. Go.
All right. Thank you. Thank you all for coming, and it's great to be here. So I'm Val McKenzie. I'm an associate professor at the University of Colorado Boulder, and I run a research lab where we study the microbes of wildlife. So what we do, there we go, is we take some of these microbiome tools that Rob was describing that he uses to study humans and human health and microbes, and we apply some of those tools to wild animals, and we're discovering a lot of new things about the microbes that live in animals. So I like to borrow and steal the human microbiome tools. This picture is showing just a few of the different types of wildlife that I've gotten the opportunity to work with over the years. So animals are home to many microbes, as you probably know, but these include all kinds of things: blood parasites, bacteria, fungi, other types of protozoans, all kinds of things. And this is normal. In a healthy animal will host all kinds of microbes, just like we do. And some of these microbes that are in animals are symbiotic partners that are probably providing benefits for those animals, and others are harmful pathogens that can cause disease. And yeah, it's a little bit like the forces of light and dark. We're going to circle back to that analogy. So an example of beneficial partners for animals includes this very cute bobtail squid. These are little squid that live in the waters surrounding the Hawaiian Islands. These bobtail squid have a light organ in their bodies that at night, when these little squid are active and hunting, the light organ is emitting light and actually protecting them from predators because it's countershading them against moonlight from above. The squid cannot produce this light on their own. They need their microbial symbiotic partners, that little green microbe. It's called Vibrio fischeri. It lives inside the light organ and has the right enzymes to be able to produce the light. And they work in this beautiful harmony to be able to adjust the light just right to match the environment. And in return, these little bacteria have a safe home that provides food for them. Another common example that you might be familiar with are the vast coral reefs all over the tropical oceans. Each little coral animal hosts little symbiotic algae that live inside their tissues. The algae are photosynthesizing and creating food for the host coral, and then the coral is providing a home for those algae. And then just like our microbes in our guts as humans that help us digest food, microbes are very important for animals for food and digestion. And so have you ever thought about how is it that a termite is able to eat wood and digest wood? It's not doing that itself. It's got all kinds of symbiotic microbes in its gut. Same for how does a bison eat grass? Or how does a vampire bat survive on blood? These are difficult diets to digest, but each of these types of animals has very specific microbes living in their gut that are capable of producing the right types of enzymes to break down those specific types of foods to provide the nutrients for those animals. But it's not all love and friendships and cozy symbioses. There's a dark side. Some of the microbes in animals are pathogens and causing disease. In Hawaii, the native birds of Hawaii have undergone drastic declines. One of the drivers of those declines is an avian malaria a malaria related to human malaria, but specific to birds, that spread through Hawaii, causing a lot of declines. Bovine tuberculosis is causing declines in ruminant animals throughout Africa. Canine distemper is spreading through African lions and causing declines. In 1994, an outbreak of canine distemper brought down 30% of the lions in the Serengeti National Park, all in 1 year. Pretty significant event. For those of you who are anglers in the Rocky Mountains, if you'd like to try to catch one of the native cutthroat trout, you might have a harder time because those native trout here are being impacted by whirling disease. Unfortunately, these kinds of wildlife disease events are increasing in frequency, and they're happening more often. We're seeing them, unfortunately, more abundantly. And why is that happening? It's related to globalization. So as people in our modern era, we're moving all kinds of plants and animals around the globe to places where they were not previously. And when we've done that, we've unintentionally, accidentally, also moved non-native microbes and diseases to new areas where they didn't belong. And then those diseases are spilling over. So for these examples I just talked about, the avian malaria was introduced to Hawaii when people brought non-native birds there. And they brought infected birds by accident. Same thing, when non-native cattle were brought into the continent of Africa, the cattle diseases spilled over to affect native ruminants. Domestic dogs, which are not native to Africa, brought in the canine distemper, which is now causing problems for the native carnivores. Same story for our trout. The non-native European trout that had been stocked in North America was the thing that brought in this whirling disease microbe. So we call this pathogen spillover. We reduce these types of events. When we have strong biosecurity, we know that we can reduce these types of events, and that's really important to think about. It's all interconnected. Human disease, wildlife disease, invasive species, all of these things are connected, and we do know how to get a better handle on it if we can work those better biosecurity policies. But how can we fight the dark side? So we've got a lot of these wildlife pathogens out here harming native biodiversity. What can we do to fight these things? In some cases, there are vaccines for wildlife, but that is really rare. There are sometimes also medications that you can use in veterinary medicine, but again, not so common in a lot of cases. So what else can we do? So last thing here is I'm going to share a little story about amphibians where we're harnessing the microbiome to solve one of these problems. And so in my lab, we work a lot on amphibians. This is the group of vertebrate animals that includes the frogs and toads and salamanders. There's about 8,000 species of amphibians on the globe right now. And so for reference, that's on par with the diversity of birds or mammals. It's a big group of animals. Unfortunately, as a group, the amphibians are declining pretty significantly. Many of the species are very susceptible to this fungal pathogen called Batrachochytrium dendrobatidis. That mouthful we sometimes just call BD for short. This skin pathogen infects their skin and interrupts their water and electrolyte balances and causes disease that can kill many amphibians. It doesn't affect people, this fungal disease. It's specific to amphibians, but it's causing a lot of declines. This guy is the Colorado boreal toad. I think he's very handsome. I hope you do too. This is a toad that is in trouble because of this fungus as well. This fungus is wiping out some boreal toads. If any of you had a chance to go to the field trip this morning to the maroon bells, this maroon bells habitat here is an exactly perfect type of habitat. This is where the boreal toads live. These are high elevation mountain toads that used to be super abundant throughout the Rocky Mountains in Colorado, and they've declined precipitously because of that skin fungal pathogen that accidentally got spread around by people. We did that too, unfortunately. So how do these toads or any amphibians protect themselves? Well, toads, like us, they've got innate and adaptive immune systems, and they can develop and evolve defenses, but that takes quite a bit of time, and this pathogen is new and spreading quickly and not giving them a lot of time. So in my work, we've studied the microbes living on these toads, and we found that some of them are really good at defending against this fungus. One of the skin bacteria we've isolated from the toads is called Xanthinobacterium. We call it JLIV for short. When you grow it in a petri dish like this in culture, it produces a bright purple color. That's a natural color of this microbe. It's called violacine. It's a metabolite chemical that these bacteria produce, and that chemical is like kryptonite against the fungus. It's a really good deterrent. So we've created probiotic baths to pour on our toads. So we go up into the mountains and we culture up a lot of their native microbes and reintroduce a bunch of native microbes onto them to protect them from this fungus. Because we're pouring this purple bacteria on them, we call it Project Purple Rain. Also in honor of the late great Prince, of course. I think he would approve, I hope. So here we are out in the mountains with a couple of my graduate students treating some young boreal toads, hoping for better futures. There's a picture in that jar there of some of the purple liquid of the bacterial juice we're pouring on them. And here are those young boreal toads now with better skin microbes to protect them against the fungus, thinking about their futures, wondering why the strange lady's pouring stuff on them. But we've got big plans for the boreal toads. We hope they're going to improve out there. So just a couple takeaways from my presentation, that there are so many benefits of microbes for animals, and they provide so many critical functions. But there's a dark side too, where there are these pathogenic microbes that are increasingly a problem for wildlife. And once in a while, when we look at things very closely, we can discover gems like JLIV in the microbiome of these wild animals to use them for good against some of these pathogens. So just a quick acknowledgement to Colorado Parks and Wildlife, who support our work on boreal toads, and the Morris Animal Foundation, and many of my lab members who've helped out with that research. So thank you so much.
All right. That was great. I have a question for you, Val, which is, you talked about all the factors affecting the microbiome, the microbes in various animals. What role does diet have?
That's a great question. Diet is really important for animal microbes, just as it is for human microbes. It's far less studied, so we don't have a lot of good examples where we know very clearly, but it's been most studied in captive animal contexts. So in some zoo environments, some animals have problems like dysbiosis, or they're having issues with their guts. And we figured out that is linked to the change in diet that they're experiencing in captivity, which leads to a change in their gut microbiome in captivity. So if you can correct the diet and microbiome issues in captivity, you can have a return to health for some of those animals.
Okay. And Rob, in people, we hear all the time about the role of diet and our microbiome and in things like sort of gut health. But just go beyond that. What is the role of diet?
Well, the diet has a huge impact on our microbes that then change what chemicals they produce, and those have impacts all over our body. So, for example, if you think about the gut-brain axis, the links between the microbiome and the brain, some of that is the microbes making molecules out of what we eat in our diet, and then those go into the bloodstream, and then they modify other things like in our brain directly. But sometimes the bacteria themselves get around the body. And then sometimes they train immune cells that then activate different things in our immune system. And as you know, there are tremendous links between the immune system and a whole range of different brain disorders. So there's a lot of different ways they're connected, not just one way. And even diet can help the vagus nerve, which is this highway of neural signals between the gut and the brain, make it signaling. And so those responses to things that microbes make out of diet, they can happen in just a few seconds after you've eaten something.
And Val, you talked about one way where, you even called it a probiotic. A probiotic was able to help a species. Is that an approach that might help with endangered animals at all?
I think there's a lot of potential for more studies of the microbiome and more thinking about how can we use beneficial microbes to help wildlife in their health or to deal with an infectious disease. There's huge potential. It's sort of an open area of study where we need a lot more people to join the science in figuring out some of those problems. But yes, I think it has huge potential.
And Rob, I'm curious about things beyond, let's move beyond the gut a little bit. You mentioned the effect on the brain. Does that go to things like Alzheimer's?
Absolutely. So there's a number of diets, including the MIND diet that's based on the Mediterranean diet, the DASH diet, and so on, that have been shown to have really good impacts on Alzheimer's and to help with cognitive performance and so forth. We're working on a large project right now funded by the National Institutes of Health that's looking at how much of that's mediated by microbes, which we know are also affected by those diets. So basically we're asking, how does the diet affect the brain? Is it by changing the microbes that then affect the brain, or is it directly, or is it some combination of those?
And related to that, what is the connection between what's happening down here and, how does your stomach talk to your brain?
Well, absolutely. Well, that's those different highways of communication I mentioned. So it's more lower down in your intestines than in the stomach, but sometimes it's small molecules that cross over into the bloodstream. Sometimes it's immune cells that migrate into the brain and activate the immune system in the brain, which is related to neuroinflammation, which is tied not just to Alzheimer's, but to depression, anxiety, autism even, Parkinson's obviously, multiple sclerosis, and a whole range of other conditions. And then finally, you have that direct signaling through the vagus nerve. So there are probiotics that you can use as effective as the best anxiolytic drugs in mice. But if you snip their vagus nerve, they stay just as anxious and just as depressed. And it's only when that vagus nerve is intact that the gut can signal to the brain by that nerve that those probiotics work for those mice. Now I should mention that these probiotics, much like the frog skin probiotics, we're not talking about something that's in yogurt or other food at the supermarket. We're talking about, for the most part, native bacteria that live in that ecosystem. And that's a really important distinction because a lot of the kinds of microbes that have really large effects, you can't buy those as a supplement because they're regulated as drugs instead.
All right. Val, I want to come back to autism in a moment since you mentioned it. But before that, I'm curious. So many animals, their survival depends on being in captivity. Now, there are lots of animals in captivity. Does that affect the microbiome?
Absolutely. We've done some studies, Rob and I in collaboration together, demonstrating the strong effect of captivity on animal microbes. And it's no wonder, when animals go into captivity, everything's changing. Their surrounding environment, so which microbes they're exposed to, oftentimes their diet changes from what they'd be eating out in the wild versus in captivity. And then they're also receiving all kinds of medical interventions and veterinary interventions and sometimes antibiotics, which all affect the microbiome. And so, yeah, we've shown that across a broad diversity of animals, from reptiles, amphibians to mammals, they've all got very different microbiomes in captivity compared to the wild. And we don't really understand all of the health implications of that. In some cases, we know that animals in captivity suffer dysbiosis, but for most of them, it's really unstudied right now.
And Rob, I think a lot of people are curious. You brought up autism. Say a little more about that.
Sure. Well, autism is one of those conditions that people have thought there might be something ongoing with the gut for a long time, just because so many people with autism have GI issues. But it's one of the diseases that's been cured in people, only a small number of people so far, but remarkable improvements both in GI symptoms and in cognitive symptoms by what's called a fecal transplant, where you basically take poop from someone who's healthy and you go in either from the top or up from the bottom to transplant it into someone who's unhealthy. And in the clinical trials that have been done at Arizona State University, it's been remarkably effective for helping both with the gut and with the brain.
Interesting. And is the thought that it's the modulating system is the immune system somehow?
Yes, that's right, and also small molecules produced by the microbiome, especially one called 4-ethylphenyl sulfate. So we've been working on that more in mouse models with Sarkis Masmanian at Caltech and other collaborators. But it's really fascinating to think that one molecule that's modified from something in your diet could affect something as profoundly as our ability to connect with another human being.
Right. And since you brought up poop, it occurred to me that I feel like I've seen a lot of things about what we've learned about animals from studying not the animals themselves, but their poop. Val?
True. Yes, we've sampled quite a lot of animal poop from all over the place.
And what can you learn?
You can learn all kinds of things about all the types of microbes that are there, if there are pathogens present. You can also learn oftentimes from a fecal sample what that animal was eating as well, which is really informative. We're not anywhere near as far along as the human microbiome work is for understanding how these things affect animals, but we take a lot of cues from the human microbiome work.
But I should say that the reverse is also true for diet. So from an animal, it's not going to answer a food frequency questionnaire or fill out a food diary, right? So the only way you can get at it is from the poop. And right now we're learning how to apply those back in humans, who it turns out are also terrible at filling out those questionnaires. So we can tell directly what you're eating from what you're leaving behind.
All right, I promised there would be time for questions from the audience, so now is your chance in the back. I think you probably should wait for the microphone. I think that's how they...
So how do we know, like, is there a test that we can do to see if our gut microbiome is healthy? Like, how do we know if we need to add certain probiotics or if we have too many of a certain one? Are there any tests that we can do so we know where we stand?
Yeah, so there's a wide range of citizen science projects like the American Gut Project that I run, as well as a lot of companies that will give you a readout of the kinds of microbes that are in your gut. Defining what's healthy is really hard, because there's this distinction between how you are right now and how you're going to be at the future. Like if you go down to McDonald's, you'll probably see a lot of teenagers who are perfectly healthy right now eating a Big Mac or something, but would you call it a healthy diet? You'd worry about what's going to happen to them down the track. So what we're doing right at the moment is longitudinal studies where we can start to understand not just does your microbiome, so the way we define healthy is usually, do people who are healthy right now have a particular microbiome? But that has important limitations. And so we're working with cohorts like FinRisk that collected thousands of poop samples back in 2002, so we can ask from the microbiome back then, what were people's outcomes, and can we not just read out current state, but predict the future? Currently there are no, there's one test for colon cancer that's based on the microbiome that's on the market. But there are very few tests that pass the FDA's criteria for a diagnostic. And you should treat this much more as an emerging area of science, where what you're going to get is information that might be interesting to you, but to be useful to you, you typically need to wait for FDA approval, which is a fairly long process.
Over there.
Yes, can you tell me any work that's being done to combat chronic wasting disease?
Oh, chronic wasting disease. Yes, this is a prion disease that's affecting elk and deer and things in the Rocky Mountains, but also across North America. I'm not aware of studies that are looking at the microbiome related to chronic wasting disease. It's probably an area that definitely needs more attention. But there are no treatments for chronic wasting disease right now. Other than, you need to isolate animals, and in the case of when animals test positive, they try to remove them from the population or quarantine them. So there are not cures for that.
As there are not for people either with those.
No, so chronic wasting disease has never yet been reported to cross over and spill over into humans. But there are other types of prion diseases known in Europe, like mad cow disease, which is also a prion disease that there are cases that have crossed over into humans. That has not happened in North America with chronic wasting, but there's a concern that because we know of other examples, maybe that's a possibility.
Over here. You, yes, you.
Interesting talk. So I'm a pediatrician, and I'm interested in hearing more about the microbiome in infancy. And I'd love to hear you comment on formula versus breast milk, and also the impact of having animals in the home, whether that be reptiles or chickens, or if you work on a farm, or it's a family that, they're farmers.
Sure, absolutely. So one thing that's fascinating about breast milk is that those early life impacts, like C-section, can be largely erased by breast milk later on. And so we've done a study with Merete Egsbo at the Norwegian Institute of Public Health, looking at those events right around birth, and then at the kids at age 8 or 10. And what we see is that a lot of the relationships with weight, with cognitive performance at age 10, those negative impacts are only seen if there's formula feeding, not breastfeeding. And Marty Blaser and Maria Goro Dominguez Bello have seen very similar things in other cohorts, so we think it's generalizable. In terms of the impact of household pets, so in general, dogs and cats seem to be protective against asthma and to some extent allergies. There's a really interesting study by Erica von Mutius, Jack Gilbert, and their collaborators looking at the Amish versus the Hutterites, where basically the Amish kids go on the farm, but the Hutterite kids are kept in a separate home environment. And there's tremendously different incidence of asthma, allergies, a wide range of other conditions linked to the microbiome, where it shows that exposure to those farm animals early on is very good in those contexts. But the data is somewhat inconsistent because it seems to depend on the population. Like in Asia, chickens seem to be protective against asthma, but in North America, that's not true. So there's still a lot to be worked out, and we really have to take a global view of these problems and look at many countries around the world to understand what's going on.
Yes. Yeah, I picked the person it's hardest to get them the microphone.
Which foods give us the most probiotics? There's controversy over dairy, and yet if you go to a grocery store, they'll have all these little yogurt drinks to boost the microbiome. Do you have any comment on which foods are best?
So any fermented food, whether it's dairy or whether it's say kombucha or sauerkraut or kimchi, any of those fermented foods will have high loads of bacteria, including things like lactobacillus and streptococcus species that are widely sold commercially as probiotics. I should note that those probiotics, there are particular strains that are good for treating particular conditions, but there's very little evidence that they'll improve your health above baseline if you're already healthy. Although things like treatment after antibiotics, that's a situation where probiotics can help you restore your gut function after that. If you're thinking about the best recommendation for foods overall to improve your microbiome, though, you want to think more about prebiotics, which are basically fertilizer for the microbes already in your gut. And what we've seen in the American Gut Project is the most important thing is not which particular food you eat, but the diversity. So, for example, people who ate more than 30 distinct species of plants in a week had much more diverse gut microbiomes than people who ate fewer species. And in fact, that was much bigger than the difference between self-reported vegans and omnivores, because it was much more important how many different kinds of plants you ate rather than whether you sometimes ate meat. So a lot of the stuff is very counterintuitive.
So a prebiotic, would you take it as a pill, or is there, I couldn't understand what you said, are there foods?
I'm sorry, prebiotics are different chemicals that you can either take as a pill or in food that are like fertilizer for your microbes. And our best recommendation at the moment is to eat a lot of different kinds of plants, which have different fibers in them, rather than to take a pill, which probably has one isolated kind of fiber that will just fertilize one kind of microbe, not all the microbes that are in there.
You had your hand up before. Row back.
Excuse me, it's like extra loud.
I think the talk has been fantastic. Two questions. One, you mentioned Alzheimer's, and I wanted to get a sense of if there are any obvious interventions and what you'd share. And the second is, I know there's a lot of contradictions. Is there any obvious thing you would do as a normal, healthy human that would affect microbiome and lifespan and life quality?
So in answer to the first question, both the MIND and the DASH diets have been well validated as decreasing the risk of Alzheimer's and improving function if you already have Alzheimer's. What we're trying to figure out at the moment is whether the reason that those diets are effective is because of microbes, and if we can tell why they're effective for one person but not effective for another person is because of differences in the microbiome that we could tell going in. And so not for Alzheimer's, but for insulin resistance and diabetes, there's a company called Day2 based on research out of the Weizmann Institute, and for full disclosure, I'm on their SAB, but they're able to give very precise readouts of what you should or shouldn't eat to improve your postprandial glucose response based on a microbiome test. So that's what we hope to be able to replicate for Alzheimer's and for other conditions. It's called Day2, D-A-Y-T-W-O. In answer, so sorry, what was the second part of your question again? Oh, interventions for normal people. So, so eating a lot of different kinds of plants. I mean, the thing is that for normal people, without that kind of test, it's all very generic, right? Like you should eat a bunch of different plants. You should eat a lot of different fermented foods. You should drink a bunch of different fermented drinks, including things like coffee and teas. You should eat nuts, like a wide variety of different kinds. You should eat a lot of leafy green vegetables. But the thing is, you know, you should avoid fries and fried foods and the hot dog stand with all the nitrates, which they don't add as nitrates, they add as celery extract, which is basically the same thing chemically. I shouldn't say that because they're probably sponsoring the event, but you know how it is. But the thing is, those are all very generic, and the problem with all of these food impacts is at the population level, the effects are small, but at the individual level, very specific things have huge impacts. And that's what we're getting towards with these individualized tests based on the individuality and diversity of your microbiome.
All right, we have time for one more question. Anybody over on the right, over my shoulder? Yes, you.
I've seen a lot of articles lately about wastewater surveillance for COVID and stuff. Are there other public health, broad societal applications of this work?
Yeah, absolutely. I'm really glad you asked me that. So the program that we have set up at UCSD, where we have 140 samplers all over campus that we process the wastewater from my lab in every day. Right now, that's specific to COVID. There's a huge number of other disorders, including respiratory viruses like flu and RSV, you can also detect in the wastewater. And what's coming is all of these things linked to the bacteria in the microbiome and other gut things. We might be able to tell at the whole city level, say, you know, how many people have Alzheimer's, how many have depression, how many have inflammatory bowel disease, all these other diseases that are linked to the microbiome. So that's an incredible AI challenge to take those signals from thousands or millions of people and deconvolute them. But there's already some very promising studies that suggest that we might be able to do it. So it's a really exciting area, bridging the individual to the population level, especially getting at the health disparities between people who can't afford to pay for an individual test or don't want to interact with a healthcare provider that they see as linked to the government, but where you could bring these benefits to everybody by city or suburb level wastewater surveillance. So it's an incredibly exciting time for that right now.
Sadly, we are out of time. I am sorry, but thank you all for coming, and thanks to our scientists for being here.
Thanks so much.